2, 1. We are here. Let's see. Oh, geez, what's going on? Oh, okay, it's working, but it's not on the live stream. So the live stream link, let's try that. Come on, live stream link. Shit, come on. It's working on its own link. It says it's live now. For some reason, it's not showing up on the main live page. Is it working? Is it working on the YouTube link now? All right. Great. I think we got it now. Are you not moving at all? Not even a little? Let's see. Oh, wait. Hold on. Let me try something. All right. What about now? All right. We're trying to get Justin patched in, so sorry everybody for technical difficulties. The Bioengineering 101 class. Your teachers are going to be me and Justin. Trying to get Justin patched in, so we're both live right now. I don't know if it's working very well. Can you guys see Justin moving at all? Or no? Justin, can you see Justin moving at all? All right. Well, then just answer questions in the chat. We'll just get started. Justin, you help answer questions that people have in the chat and stuff like that. Sound good? All right. Okay. So Bioengineering 101, if you go to the Google Classroom, we have the syllabus. We're going to go over the syllabus really quick and just get everything going. So all the videos for this class are recorded and they can be found on the Odin YouTube channel. So please go to the Odin YouTube channel if you miss a class. It's just youtube.com/theodininc, and you should be able to watch any video that you missed out on. So don't worry about that too much if you can't make every class and you miss a video. This course is no experience required. We're going to teach you everything you need to know. So don't worry about that stuff too much. We're going to tell you about everything from what DNA is, how to use all the equipment that you received in the kit, and basically get you to the place where you can do genetic engineering by yourself without any outside help from anybody else. Some of the goals and things we're going to do in this course is how to use pipettes, how to make agar plates, how to use sterile technique, culture bacteria, genetically modify bacteria, and even go over CRISPR and things like that. So the textbook for this is Molecular Biology of the Cell. There should be a copy of Molecular Biology of the Cell in the Google Classroom under week 1. So just go and download that from the link and follow along. So at the end of the syllabus, it tells you what weeks we're going to cover what chapters of molecular biology of the cell. So week 1 and 2, we're going to cover chapter 1 and chapter 2, right? So check that out. Try to keep up to date with that. There's going to be a lot of reading and a lot of work and stuff with this class, but try to do the best you can. Um, I understand that you might not be able to uh, get all the reading done. It's a lot of reading, but just do the best you can, right? Because the class is just going to be about subjecting yourself to this information over and over and over again, right? Like that's how you learn. Somebody tells you about biology and certain things, and you just keep hearing it and you keep reading it and you keep learning it, and the information just builds and builds and builds up. That's the way you're going to learn how to do all this stuff in the class. Um, so yeah, do your best, the best you can with the readings. So there's 2 programs that we're going to use and go over in this class. One is SnapGene. So get the SnapGene viewer. It's free. It helps you look at DNA. We're going to go over that, how to look at DNA and analyze DNA and stuff like that. The other is VMD. It's Visual Molecular Dynamics, I think it stands for. It allows you to look at proteins, look at the 3-dimensional structure of proteins, and all that good stuff, because it's an important part of molecular biology, biochemistry, genetic engineering type stuff. So each week, the first thing you should do is go to the Readme. The Readme will have the information for the week for the class, what things you're going to kind of go over, what papers you can try and read. The papers, obviously, reading are optional, and some of the things you will learn this week. So this week, being week 1, we're going to start out learning how to use a pipette and how to use a scale. So that'll be the experiments for this week. These things are the, building blocks of doing hands-on molecular biology and genetic engineering. So if you don't know how to do these things, even if you kind of think you know how to do these things but aren't completely sure, make sure you reinforce those skills by performing the experiments. It never hurts to perform the experiments. If you've been working in a lab for 10 years or something like that, then maybe you can get away with not performing these things, but, it's always a good idea to go over these skills and experiments, to brush up on it so you know what's going on. If you have any questions, please try and post them in the Google Classroom. That's the easiest way for the instructors to answer the questions, and other people can also gain and learn from your questions, so try and post them in the Google Classroom. The live streams of this class are going to be every week at 4 p.m. Pacific time, California time. So try and make it out for these live streams. We're here to answer any questions you have, help you with experiments, give you advice on your love life, whatever you need. You just come to us. We're here to help you. All right? So does anybody have any questions right now about anything that's going on? Anybody? Any questions? How are things going? Is everything going good? Yes? No? All right. Great. Sounds good. All right. So the first week, what we're going to learn about is we're going to learn the first and second week about cells. Cells are a really interesting phenomenon, right? Because cells, all the cells that we know of, only come from other cells, right? So there are no cells that we know in existence that haven't come from other cells. And that kind of, you know, blows the mind. We can't recreate cells from scratch, not yet at least. Um, the only thing we can do is replicate cells and modify those cells that we replicate. Um, and It makes you think a lot about where the first cell came from. Where the first cell came from, how is it able to do what it does? Some people are, you know, under the impression that maybe the first cell came from an asteroid or outer space, another planet somewhere, and landed on Earth and seeded the planet through panspermia. Other people think that, you know, cells just developed through evolution. Lipids that formed the cell wall just naturally formed into this, you know, lipid micelle shape and encapsulated DNA and proteins and, you know, organisms were born. But honestly, you know, we really don't know. We have no idea where cells came from at all. No idea. So, you know, that's something that people need to figure out. I know there's a bunch of people right now. It's starting to become really popular and trying to create synthetic cells. Like try to create a cell from scratch. That's going to be really interesting when that first happens. They've tried lots of different ways to figure out how to do this. It's just cells are complicated. They really are, right? The cell is composed of the cell wall, different organelles inside the cell, DNA, proteins, different molecules, small molecules, electrolytes, salts, all these different things. Having them in the cell is it's difficult. And not only that, like cells are all different, right? So like a human cell is much different in shape and size than a bacterial cell. Even bacterial cells are different between each other, right? They're different in shape and size. Now, bacterial cells are generally much, much more difficult to see under a microscope than human cells or plant cells or yeast cells. These cells are, you know, maybe almost 10 times bigger than bacterial cells. Um, and bacterial cells are really nice. Through this course, we're going to work mostly with bacterial cells. And that's because bacterial cells are quick to replicate, right? So the amount of time it takes for a bacterial cell to replicate is like 20 to 45-ish minutes. So what you can do is you can grow bacteria and grow it fast. So when you're working in molecular biology and genetic engineering, you could get the results from your experiments pretty fast. Whereas yeast takes it takes, you know, a few days, and plants take, you know, tens or months, tens of days or months. Human cells are kind of intermediate between bacteria, I mean, yeast and plants, in which if you're trying to genetically modify some human or mammalian cells, it can take, you know, 5 to 10 days ish or something like that, right? But yeah, this course focuses on bacteria because it's small, it's easy to grow, and understand all the bacteria used in this course is not hazardous. It's not harmful. So you don't have to worry about that at all. The bacteria won't kill you no matter what you do. You can eat the glass vial that the bacteria is in, and you're probably not going to kill yourself. Not saying you should eat it. It's not a good idea, but you know, all this stuff is pretty safe, so don't worry too much about it. Bacteria gets a really bad rap, right? When you think about it, it's like less than 1% of the species of bacteria that actually cause any sort of illness or disease, right? So, you know, understand bacteria are the workhorse of molecular biology and genetic engineering. If you're going to do molecular biology and genetic engineering, you're going to have to learn to work with bacteria. It's just the way it is. Because it doesn't matter how hard you try to, to avoid it, you're going to have to use it for purifying DNA or expressing a protein or doing some experiment. Bacteria is going to be there. So, get used to working with bacteria, and that's the reason we do it. So, basically, all living cells have DNA. Now, I know some people might be like, well, what if you say a virus is alive or something like that? Not all viruses have DNA, and that's true. Not all viruses have DNA. Some of them just have RNA. But if we say just like all viruses aren't living because they need a host, then all living organisms that we know of have DNA. And they usually go about the process of replicating their DNA and making proteins all in the same or very similar ways, right? So inside the cell, when organisms are replicating DNA, the proteins involved are similar. A lot of things about the process are similar. It's very similar in many different areas. So, despite cells having, different shapes and sizes and different size of genomes and different amounts of DNA, generally they all abide by very similar principles of function, using, channels and using DNA replication and protein translation and all these different things. We'll learn more about these things in the class, and if you read the Molecular Biology 101, it'll have, or Molecular Biology of the Cell, it'll have a lot of information on these things, right? So don't worry too much if you don't know a lot about it, but go to Molecular Biology of the Cell. It's free. You could search it on the internet and find it, Molecular Biology of the Cell. It's a book that you're not going to need to know everything in, but generally at one point of time or another, the information in that book is good to have. The knowledge in that book is good to have, and it'll be very beneficial to you as you pursue a career in molecular biology and genetic engineering, right? So the things that go on in these cells happen because of proteins. So DNA turns into RNA, turns into proteins. They call this the central dogma of molecular biology. And proteins are these little machines that keep cells alive. If you go on like YouTube or something, let's see if I could find a YouTube video about proteins moving around, and then you could see what's going on. And like proteins are actually super cool. So you have to understand that like DNA and proteins are they're just these molecules that kind of flop around inside cells and do things, right? And them flopping around and doing things is basically what keeps you alive. Let's see. What do we got? Trying to find a protein simulation. Let's see. All right. I don't know if this is gonna show up well. Let's see. So yeah, you have to imagine that proteins are these little small, tiny things, nanometers in size, that move around, right? They're in water and they're fluctuating in water and moving around and waiting for stuff to happen, trying to figure out what they should be doing. So I guess this is protein folding simulation, but it'll probably be interesting anyway. All right, let me see if I can pop this window out. All right, let me add in a window capture. All right. So what we're looking at here is we're looking at a simulation of a protein. So basically what somebody did was they just calculated Newton's laws of motion for the different atoms and molecules in a protein. What a protein would be like if it was in water. And this is kind of what it looks like. It's just kind of flopping and moving around and doing stuff. But this moving around, right, its ability to move around in water gives it machine-like properties. So then it can actually do things, right? It can bind to different things. It can, release different things. It can cause chemical reactions on different things. But yeah, that's basically what a protein would look like if a human were looking at proteins. So proteins are little machines made by DNA and help cells do their thing. They help cells, stay alive, grow, function, whatever. All right. So even though, cell sizes of human cells are bigger than bacterial cells and human genomes are bigger than bacterial genomes, don't make the assumption that like a cell size or a genome size, the number of bases in the DNA of an organism, correlates with the the complexity of the organism, right? So humans don't have the largest number of genes or the most complex DNA or genome of all organisms, right? So they might have a larger number of genes than bacteria and stuff like that, but they don't have the most genes. Some organisms, like plants, have so many, they've gone through genome duplication events, so they have so many different copies of their genes, right? So humans have 23 different chromosomes, and we have 2 pairs of each chromosome. So we have a total of 46 different chromosomes, and then our sex chromosomes, right? So some organisms have like 10 pairs of chromosomes and stuff like that. And they're way more primitive than humans. They have way more genes than humans, even. So, in a lot of modern science, especially medicine, you see a lot of stuff about how, like, mice and rats and fruit flies and all these organisms are used to try to replicate humans. Now, you have to understand that on a cellular level, the level of the immune system and metabolism and all these different things, these organisms are so much different than humans. So when people tell you that they've like cured autism or something in mice, don't believe them at all, right? Be very suspicious of this stuff because cells are different, right? Like we have so many different cells and we have a different immune system and we have different metabolism and all these things than all these other organisms. Is it a good starting point? Sure, but the chances that it translates easily and directly to helping humans is not good. It's generally not good, right? And we're not really allowed to experiment on humans. So we're left in a very, very weird area where scientists kind of have to experiment on mice and fruit flies and stuff. I don't think we need to experiment on fruit flies, but don't tell anybody I said that. Might piss off a few scientists out there. But I'm just saying, like, you see some of these fruit fly research, and they're like, we got some fruit flies drunk, and then we tried to make them climb up a wall or something. And you're like, all right, like, I kind of get it. You're trying to, like, extrapolate that to humans and do science, but, like, how a fruit fly responds to alcohol is probably not going to be extremely similar to how a human responds to alcohol. So maybe we could figure out something else you can test or work on, or just something in general. So we all hear about DNA and all this different stuff, and people say DNA is made of A, T, C, and G, right? And we hear that you got a cell and you got cell wall and lipids or proteins and all these things. And I know these things are like hard to visualize and understand, but what you have to start to understand is that these macromolecules, these big molecules, they are basically composed of 5 different things. So things in the cells are composed of about 5 different things, and that's hydrogen, carbon, nitrogen, oxygen, and phosphorus. Now, there are other things like sulfur and whatever, that are also involved, but these are the 5 major things. So when you think about DNA, when you think about proteins, what it actually is it's atoms that form molecules, and these molecules have some sort of structure. And this structure and the characteristic of these molecules, they contribute to how things work, right? Like carbon, carbon is a fairly neutral atom. And by neutral, you mean that the charge that it has based on the electrons and its atomic orbitals is fairly neutral. So it's probably not going to attract or have a positive or a negative charge much at all. Certain situations, when you have like a carbon ring, you can start to see weird, strange effects. But in general, carbon's just going to be kind of neutral. Nitrogen, it's going to be more positively charged. Oxygen, negatively charged, right? So the composition of these molecules with the positive and the negative charges and the number of these atoms and the arrangement of these atoms in the DNA and proteins, that's basically what gives these things function, right? These little atoms and molecules in the DNA or in the protein that have specific characteristics like a positive or negative charge. And these positive and negative charges, when you put them together in groups and other things, then they have different forces, different abilities to do different things. right? So, for instance, if we look at DNA, let me see if I can pull up a picture of a DNA backbone. Where did that go? All right. Not there. Right. No. Almost. So DNA is composed of two strands, right? And these two strands they interact with each other and they're held together by what are called hydrogen bonds. And these hydrogen bonds are basically an interaction that is based on the fact that certain molecules have different charges. And those different charges allow the molecules to interact, right? So let's see, where is this? Sorry, I don't know why I can't find this in molecular biology of the cell. I thought I had it already set up. So I'll just blame myself. No need to blame me. Where is... maybe I'll... Oh, here we go. All right. All right, let me share this on the video. So you all can see it. So all right, here we go. So here's a picture of DNA right here, right? And you have DNA has two different strands. And these strands of DNA, you could see these 2 and 3 different lines between the two things. Those are representing hydrogen bonds. And these hydrogen bonds are just ways for DNA molecules to interact, right? The nucleotides that were formed wherever you believe they were formed, in the early hot springs on Earth or wherever, these molecules came together because they were able to interact, positive and negative charges and things like that. And this is basically how all life interacts. So how all protein molecules, how all lipids, and how all everything works is it uses these positive, negative, and other charges to do stuff. Right? So cells specifically, overall, they even have a voltage, right? Now, they don't have voltage in the sense we normally think about it, where like a cell can shock you or something like that. What they have is they have a difference in voltage potential based on the amount of different ions that are inside versus outside the cell. Right? Because these cells, they also use these positive and negative charges and things to function on a larger level by producing things like action potentials, right? That help our nerves and neurons communicate through our whole body. So like on the most primitive level, we're all just a bunch of neutral, positive, and negative charges doing things. All right. Any questions on something? Any questions? Any questions? Anybody? You got questions? Let us know. Me and Justin are here to answer your questions. I know it's crazy. Don't don't freak out. Like I was talking about, just go over molecular biology of the cell. Go over the book. I know it seems crazy, and all this stuff is just like, what? I don't know. What's going on? What's DNA? What's a protein? Just read molecular biology of the cell. Find time, read some of it, start to take this information in and understand this information that we're throwing at you. A lot of it is just going to be keep bombarding your brain with the information, right? Because it is kind of like learning a whole new language. It completely is. All these different terminologies and all these different things, it's just like learning a whole new language. So don't worry too much if you don't understand, and feel free to ask questions. There really are no stupid questions. We're here to help answer all your questions. So just let us know what you got. All right. So cells. So the 2 experiments we're going to do this week that'll help us next week when we start to get into actually working with cells is how to pipette and how to use a scale, right? And these things are going to be essential in your molecular biology career. Now, if you go to week 1, we have the experiments, and we should have tutorials with pictures and directions. Mess around with your pipette a ton. It's just going to take time to get to learn to use it and figure out how to use it, and understand that a pipette is semi-precision scientific instrument. So be gentle with it. Like, try to get a feeling for it. Don't try to do things fast. Do it slow and do it over and over again until you really feel like that you have a grasp. A lot of techniques in biology you're going to learn are pretty physical. They are actually like hands-on experiences and experiments that you're going to have to do. And the only way to learn how to do that is by, practicing and doing it. It's kind of like picking up the guitar, right? You can read about it a bunch, and you could read how to play the guitar and try to understand how to play the guitar by reading and watching videos about it. But the only way you're really going to learn how to play the guitar or become good at playing the guitar is by actually, picking it up and playing it. And it's going to take a little bit of time. But over time, you're going to start to become good at it, and you're going to have experience with it, and you're going to be fine. So this week, first thing you're going to do is learn how to pipette. And the other thing you're going to do is learn how to use a scale. Scales are, generally pretty easy, simple to get a hold of, get a grasp on, but still, it's just helps you understand what you can do, avoid common mistakes and pitfalls that you might not have thought about if you haven't really used a scale all the time. Just so that you can do the future experiments, because in future experiments you're going to have to measure out stuff and mix stuff and things like that. It's something you'll always use for the rest of your life and the rest of your career in molecular biology. I use a scale almost every day. I'm sure Justin does also. Yeah. Yeah, maybe we can hear you, Justin. I can hear you. I don't know if they can. But we'll figure that out next time. Don't worry. We're going to make sure that I don't know if we'll have two instructors on every video, but we'll figure it out. It'll be all good. I just wanted to introduce everybody to Justin. I guess you can only see his picture kind of there. Um, but I'm sure you could find him online somewhere on like Facebook or LinkedIn, or are you on some social media, Justin? Yeah, so he's on Twitter, Instagram, and all the usuals. Um, so find him there and connect with him. And, like I said, try to keep your questions to the Google Classroom if you can, because other people can benefit from them instead of just contacting us individually and asking us about this stuff. But otherwise, that's it on cells for this week. So, does anybody have any questions about cells? Any interesting thoughts on cells? Um, Make sure you do the two experiments so you're ready to proceed and do more experiments next week. But if you have any questions, let us know. If there's no more questions, we will sign off and see you all next week. So we're going to be doing this at the same time, same place. We'll try to make sure there's no technical difficulties next week. I don't know what it is with these streaming platforms. There's always something. It's always something. I can never just get it correctly the first time. All right, everybody. Well, Justin and I say see you later. We'll talk to you next week, and let us know if you have any questions.